Hello students, this is a lecture on Chapter 1, Development and Structure of Cells and Tissues from the Chiego Histology and Embryology textbook. The cell structure and function. The cell consists of a nucleus, cytoplasm, and its function is absorption, assimilation, respiration, irritability, conductivity, growth, reproduction, and excretion. The fundamental structures of life, DNA and RNA, are contained in the nucleus of the cell. Cells are the smallest living units capable of independent existence. The plasma membrane or plasmalemma is the cell membrane that envelops the entire cell and provides a selective barrier that regulates transport of substances into and out of the cell. Included in the plasma membrane are ion channels that can allow certain chemicals to penetrate while keeping other chemicals out. Common cell characteristics include a cytoplasm and a nucleus. A nucleus is found in all cells except for mature red blood cells and blood platelets. DNA contains the genetic information. RNA carries information from the DNA sites of protein synthesis in the cell cytoplasm. The cell cytoplasm contains structures necessary for the absorption and for creation of cell products. Lysosomes are present in all cells except red blood cells, but they are prominent in macrophages and leukocytes. They contain a variety of acid hydrolases which function in breaking down substances both inside and outside the cell. The mitochondria are a major source of adenosine triphosphate, or ATP. Therefore, they are the site of many metabolic reactions. They generate energy and are a major source of ATP. Centrioles are short cylinders appearing near the nucleus that migrate to opposite poles of the cell during cell division. Microtubules probably function as structural and force-generating elements and relate to centrioles in relation to mitosis. The cell cycle is the time required for the DNA to duplicate before mitosis. Watch the posted YouTube video on the phases of mitosis for more information. Meiosis is the process of reduction of the number of chromosomes to half the normal number in the germ cells or sex cells, such as eggs and sperm, to allow fusion of the male and female germ cells. For more detailed information, view the video lecture on meiosis. Cells do not last forever. They have a limited lifetime, and the programmed cell death is referred to as apoptosis. Here are some definitions for aid in understanding. Induction is the process in which an undifferentiated cell is instructed by specific organizers to produce a morphogenic effect. Differentiation is the process by which cells acquire individual cellular characteristics from an undifferentiated state, that is, specialization. Growth factors are chemical substances that induce cells to initiate specific cellular processes, including DNA synthesis. The three periods of growth are the proliferative, the embryonic, and the fetal. The origin of tissue begins with fertilization of the egg or ovum, which occurs when sperm contact the egg in the distal part of the uterine tubule. The fertilized egg then grows and is termed the zygote. Watch the video on fertilization and implantation included in the module for more information. On the dorsal surface of the embryonic disc, the ectoderm forms the neural plate, whose lateral boundaries elevate to form a neural tube that will become the brain and the spinal cord. The endodermal cells also form a tube, which become the gastrointestinal tract. Tooth enamel is derived from ectoderm, whereas dentin, pulp, cementum, and periodontal ligament derives from mesoderm. Developmental abnormalities or defects may be caused by environmental or hereditary factors. The 3rd through the 8th week of embryonic development are the most critical time because this is the period of differentiation. The fetal period from 8 weeks to birth has a declining susceptibility for developmental problems. This concludes this lecture on Chapter 1, Head and Neck, Histology and Embryology.